All right, so we are going to go over the second half of our notes for photosynthesis. So we just talked about in our last video about the light reactions, and now we're going to talk about the dark reactions or the Calvin cycle. So make sure not to get confused because those names are interchangeable. Calvin cycle and dark reactions mean the same thing. All right, so let's start out with our reactants. Remember that reactants are always the things that are on the left side of the equation. And for this, we have our 12 ATP and our 12 NADPH that were made during the light reactions. So we definitely need those. And then we have our 6 carbon dioxides and our 6 RUBP. And it's super important to remember how many carbons each of these molecules has, because otherwise, it's hard to do the math and understand and make sense of all of this. But when we know that we have 6 RUBPs and each of those is 5 carbons, we know that we have a total of 30 carbons. And so that'll come into play in just a little bit. So our products, which are going to move to the right side of our arrow, our equation, we're going to make a glucose. So that's a C6H12O6. And then we're going to make 6 RUBP. So a lot of times you don't see the same thing on the reactants as you do on the product side, like we do here with the RUBPs, but we need RUBPs to be made so that we can just keep this cycle going and continue it. So it's really important that that happens. And this is all going to take place in the stroma. So if we remember back to our light reactions, the thylakoid is where our light reactions takes place. And that's because the chlorophyll is in there. So the light needs to reach the chlorophyll to excite it and to make all of those steps occur. And then once we've made our 12 NADPH, it's going to move out into the stroma and we'll make our 12 ATP too. So this takes place in the stroma. All right, so here's our whole little chart. We're going to go over each of these steps one by one. So don't feel overwhelmed by looking at this, because by the end, when we pull it up again, you should understand kind of what everything means. All right, so let's start. Our first step is carbon dioxide fixation or carbon fixation. And this is basically where our carbon dioxide that we started with and our RUBPs, which we also started with, are going to meet up and they're going to combine to basically carbon can donate its carbon. Carbon dioxide can donate its carbon. So now with our 5 carbon molecule of RUBP and the 1 carbon from carbon dioxide, we now have 6 available carbons and we have a total of 6 of them. So 6 times 6 is going to be 36 carbons available from this right here. So 6 carbon dioxides meet up and combine with 6 RUBP. That gives us a total of 36 carbons. And when we make PGA, PGA only has 3 carbons. So 36 divided by 3, and we will get 12. So we end up getting a total of 12 PGAs, PGA each with 3 carbons. Okay, so we have 1, we have 5, and then we need a total of 6 here. So 2 of the 3 carbon molecules. And then down here, this is our total. So we have 6 carbon dioxides, 6 RUBPs, and if we did the math there, that's 36, and we need, we divide by 3 and we get 12 PGAs. The problem here is that PGA is a 3 carbon molecule and it is unstable. And this is not scientific, but I'm just throwing out some things to help you remember. It's missing the love of its life. It's missing what completes it, okay? So PGA is unstable, not happy. It's looking for more. It's looking for something. So the 12 PGA that we just created from the RUBPs and the carbon dioxide are going to combine with our 12 ATP and our 12 NADPH. Think to yourself, where did these two come from? They're very familiar looking. Where did they come from? Okay, so the answer is the light reactions. These were our 2 products from the light reactions. So they combined with our 12 3 carbon molecules to make 12 PGAL. And I've been joking that this L stands for the love of its life. So PGA now is PGAL because it found the love of its life. And the phosphate, 1 phosphate is gone from the ATP. So we now have 12 ADP. And NADPH, our little taxi, dropped off its H. So now we're left with 12 NADP instead of NADPH. Okay, so NADP and ADP, they have done their job, and now they're going to go back to where they came from and do it all over again. But we're done with those guys for now. So NADP and ADP go back to the light reactions in the thylakoid so that they continue these cycles. Okay, and so you can see on our little chart here, that's all happening right here, PGA, and it's going to turn into the molecules of PGAL. And we have a total of 12. I know this says 2, but it's just trying to use 1 as an example. But we have 12 total PGA, and then we move to 12 total PGAL. All right, so we know that PGAL is a 3-carbon molecule. We're going to combine 2 of those to make 1 glucose. And you see here how the math adds up. We have 3 carbons plus 3 carbons gives us 6 carbons. We have 6 H's plus 6 H's gives us 12 H's. And 3 oxygen plus 3 oxygen gives us 6 oxygens. So we've made 1 glucose out of 2 PGALs. But I said we had 12 PGALs. So how many does that leave us? We used to, 12 minus 2 is 10. So we have 10 PGALs left. PGALs have 3 carbons each. So 10 times 3 equals a total of 30 carbons left. Luckily, this all works out. So we are able to reform our 6 RUBPs. Each RUBP is 5. We need 6 of them. 6 times 5 is 30. So we have just enough carbons to make our new RUBPs. These RUBPs are going to go back to the very beginning right here. So we made our organic compound, our glucose, out of 2 PGALs. The rest of them are going to form the new RUBPs so that everything can start back over. And then seriously, it's a cycle. It just keeps going and just keeps going. Okay, so here we have all of our steps. This should all make sense to you now. And then we have some factors that affect photosynthesis. All of these things that are in these equations, if you change them, it would obviously change something in the equation. So the amount of water that we're putting in, the temperature, the light intensity, how many photons are being received by that chlorophyll, the amount of carbon dioxide, that's on our reactant side. All of these things are going to affect it. So there are some special adaptions, and C4CAM photosynthesis, this happens in tropical areas, areas where it's really hot. Basically, it's going to affect that leaf mouth, so that stomata, from opening and closing at certain times. So the stomata is going to want to stay closed while it's really hot outside so that all the water doesn't just evaporate and leave, right? And so it's gonna control that stomata and allow it to work as efficiently as it can for these plants. Some videos, this PowerPoint is online, so you're more than welcome to go watch those if you'd like. But if you have any questions, then just email me, and good luck studying.